Right. So ELPHDIUM, it's a type study we have in our syllabus. So its another name is ELPHDIUM polystomella. So due to many chambers present in its, the shell. Right. So here, what is ELPHDIUM? ELPHDIUM belongs to phylum Protozoa. It is a foraminiferum. It's a rhizopodian protozoa. Okay. So the most common shelled protozoans are foraminiferans. So belonging to the order Foraminiferida. Okay. So you remember the order name. This is somewhat important for the examination also. And then yeah, their shells are typically many chambered and perforated. Perforated means having pores. Randralu undadam annam. Okay. So when they die, their shells reach to the ocean floor and contribute to the formation of bottom sediments. They are called foraminiferan ooze. Okay. So now our topic is ELPHDIUM crispum. ELPHDIUM crispum. So which we have seen the diagram. Just I'll show some morphological features of ELPHDIUM. There is a problem with this. Yeah. So this is the shell we are observing. Okay. Here the ELPHDIUM contains many chambers. Actually, just like amoeba, entamoeba, and paramecium, the actual animal we cannot see in the diagram. Okay. The animal is nothing but its cytoplasm. The cytoplasm is covered by this calcium carbonate made shell. Okay. Anyway, here the shell of the ELPHDIUM. Okay, it is smooth at the edges of the shell. That is called generally keel. Okay, and the center point, middle part of the uh, shell, it looks like a rounded structure called umbo. It's not a chamber. In the middle of the shell, there is a rounded elevated structure called umbo. Okay, and if you observe these shells, there are many pores, randralu untai. Okay, through which, so these the pseudopodia are emerged out. The pseudopodia are nothing but the cytoplasm. The cytoplasm oozes outside in the form of pseudopodia as they are interconnected with each other and looks like a network or net-like appearance. So such kind of pseudopodia are called reticulopodia. Okay, so yeah. So in each chamber, if you observe, there is a concave anterior concave surface and posterior convex surface. At the edge of the concave surface, if you observe carefully, there are some structures, elevated structures with cytoplasm. They are called retinal process. Okay, so they also contain pores from which the cytoplasm comes out. And each chamber is interconnected with these pores. Okay, so this is the diagram. And then let's see the points. Yeah, so if you observe the classification we all know, it belongs to protozoa. Subphylum is Sarcomastigophora, and super class is Sarcodyna, and the class is Rhizopodia, and the subclass is Granuloreticulosa. Order is important, that is Foraminiferida or Foraminifera, you can also call it as. Okay, then where it is present, it is completely marine organism. It is not parasite. So the problem. Yeah. So it is a free living, it is not parasite, and it is a marine organism. And generally, it lives in the bottom area of the sea. So about 600 meters depth it lives. So yeah, littoral zone of the sea. It is commonly found creeping on seaweeds. Seaweeds paina adi move అవుతూ మనకు కనిపిస్తుంది, just like our snail, but very smaller in size. So then just now we have seen some structural information. Yeah, here this is another diagram of ELPHDIUM in different views. Okay, so ELPHDIUM is a dimorphic form. Here I'm giving most of the points. Here it appears in two forms. One is microspheric form, another one is megalospheric form. Okay, so microspheric form, when compared with the megalospheric form, it is slightly smaller than the megalospheric form. Okay, so here in microspheric, here the diagram is not given correctly. Actually, this is microspheric, this is megalospheric. It is bigger in size, it is smaller in size. It contains, megalospheric form contains only one nucleus. This is not correct. Okay, so this is microspheric form. It is a multinucleated phase. Anyway, so just now I told you about umbo. Okay, so the terminal part is smooth, so that is called keel. Okay, so we will see what is alar processes. Okay, so at the base of each chamber, so an elevated structure like structures are present, that is called alar processes. Anyway, so this is just diagram. Now let's see the points. Yeah, ELPHDIUM crispum is a dimorphic form. One is microspheric form, another one is megalospheric form. So you should remember this. The microspheric form is smaller, multinucleated. That means it contains many nuclei, and with a smaller initial chamber. The first chamber of the microspheric form is smaller, and it performs asexual reproduction. You should remember this is very, very important. And in the same way, exactly opposite, the megalosporic form is large, uninucleate with a larger initial chamber, and it performs sexual reproduction. Okay, so you should remember these two. So total chapter depends on these features. If you remember these features, okay, there is no need to worry about the ELPHDIUM. Okay, so then shell. The shell of ELPHDIUM crispum is small, translucent, not transparent, not opaque, and pale yellow in color. The shell consists of mainly calcium carbonate. idokati important point we should remember. So the shell of ELPHDIUM is made up of calcium carbonate. Okay, and some amount of silica and magnesium sulfate are also present in its structure, but it is mainly composed of calcium carbonate. Remember. So why it is called as polystomella? Because the shell is polythalamus or multilocular. That is, it consists of many chambers. చాలా గదులు ఉంటాయి కాబట్టి, so as it contains many chambers, so the shell is called polythalamus. And you should remember that if you can understand this, just now we have seen the lateral view. So it is coiled in a plano spiral manner. That is, all the coils lie in the same plane. Okay, central axis chuttu, they form whorls or whorls. Okay, one above, just like a multi-storied building, but in a twisted way. So it is that is called plano spiral manner. Just wait, I'll show the diagram again. So here actually many rows or lines are present. That arrangement is called spiral arrangement. Okay, plano spiral way they are coiled, all the shell chambers. Okay, so all the chambers are filled with cytoplasm. You should remember one thing, that unlike amoeba, paramecium, trypanosoma, or any other protozoan, it doesn't contain the plasma membrane or pellicle. It contains only shell. So that's why it is also known as naked animal. Okay, yeah, shell walls of animal bears pores, randralu untai shell paina. Okay, through which the cytoplasm continues from one chamber to another and also comes out forming pseudopodia. Okay, cytoplasm comes out of these pores and forms pseudopodia, which are reticulopodia type. The concave posterior surface bears retral processes. Okay, just now I told you in the diagram, filled with cytoplasm. Okay, peripheral part of shell is rigid and consists of continuous rim, like a smooth structure. So that is called, keel. Okay, so, and whereas the central part is rounded, that is called umbo. Just now I have shown. And this is important point, you remember, trolaculum. Simple, the first chamber of shell is called, initial chamber of the shell or the ELPHDIUM is called trolaculum. Trolaculum, you remember. The first or initial single chamber is called trolaculum. You should remember that. Yeah, as the animal grows in size, successive chambers are laid down in a spiral manner, forming orals or, stories like. Okay, so each chamber is larger than the preceding one. Okay, so and each new oral partially overlaps and conceals the older one. అందుకే మనకి కొంచెం జస్ట్ ఎక్స్ట్రా కనబడుతుంది. ఓకే, సో ప్రతి స్టోరీ, ప్రతి వరుస కూడా దానికన్నా ముందున్నదాన్ని కొద్దిగా ఓవర్లాప్ చేస్తుంది. Okay, the overlapping portions of the chambers are referred as alar processes. Okay, overlapping chambers of portions of chamber are slightly elevated. They are called alar processes. You should remember there is a difference between retral processes and alar processes. Okay, overlapping portions are alar processes. Okay, whereas the concave surface of each chamber contains smaller elevations. They are called retral processes. Yeah, cytoplasm as usually. All the shell chambers are filled with a mass of inner cytoplasm. Besides a thin layer of outer cytoplasm, యా సో ఇక్కడ మనకు ఇంకొక విషయం ఏంటి అంటే ఆల్వేస్ సైటోప్లాజం కంప్లీట్లీ కవర్స్ ఇట్ కమ్స్ అవుట్ అండ్ ఇట్ సరౌండ్స్ ది టోటల్ షెల్. దట్ ఈస్ ద పాయింట్ యాక్చువల్లీ. అండ్ దెన్ yeah, so there is an inner endoplasm and outer ectoplasm as usual. Endoplasm contains nucleus or nuclei. న్యూక్లియస్ ఎందులో అంటే మెగలోస్పెరిక్ ఫామ్ లో. డయాగ్రామ్ మిస్టేక్ వచ్చిండు. ద డయాగ్రామ్ okay, so nuclei are present in microspheric form. Microspheric form is a multi-nucleate form. Form is a multi okay multi-nucleate form. Food particles, minute vacuoles, Golgi bodies, all the cell organelles are present in the endoplasm. Okay, and ribosomes, sorry, some color granules are present. They are called xanthosomes. Okay, their function is not clear. Okay, then nucleus. The inner cytoplasm or endoplasm contains a single nucleus in megalospheric form and many nuclei in microspheric form. Okay. So then reticulopodia, I have already told you about. So reticulopodia are nothing but the pseudopodia of ELPHDIUM crispum are thread-like branching and anastomizing. That means they overlap with each other to form a network-like structures. They are called reticulopodia. You can also call them as, myxopodia. Okay, each consists of two regions, an inner fibrillar axis. So how the cytoplasm simply that much sharp, just like a rod. So due to presence of inner fibrillar axis and the outer fluid-like cortex is present in each, the pseudopodium. Reticulopodia help in organism in locomotion, nutrition, and the construction of shell and cyst walls. So during harsh conditions. Yeah, nutrition is not much important, but this question was asked once. So what is the kind of nutrition in most of the protozoans? Holozoic. That means simply eating solid substances. So Elphidium crispum is also holozoic. It exhibits a holozoic nutrition. It feeds upon minute organisms like diatoms, other smaller protozoans, crustacean larvae, etc. Digestion is normally completed outside because the digestive secretions from the cytoplasm, they are secreted outside. Okay, outside the shell, and the products of digestion pass into the endoplasm. After digestion, the products are sent to endoplasm. So now the reproduction or life cycle. It is very, very simple. It exhibits a clear-cut alternation of generation. So in which from the microspheric form to megalospheric form, from megalospheric form to microspheric form are produced alternately. Okay, so now let's see. For example, see this. This is sexual reproduction part of life cycle. This is asexual reproduction part of life cycle. For example, if you see this, this is the megalospheric form, which is adult megalospheric form having single nucleus. ఇది ఒకటి గుర్తుంచుకుంటే we will remember this life cycle easily. So, Right. So this megalospheric form, okay, it produces gametes, two different megalospheric forms of same species. Okay, they produce isogametes. Okay, each one with a pair of flagella. Biflagellate isogametes are produced from adult megalospheric forms, two different animals, two different Elphidium species. Okay, but both are similar. Okay, this one is produced from one animal, this one is produced from another animal, not from this. Okay, so they are just like male female gametes, but they are similar in structure and shape. Okay, so these gametes are released into the water, the fertilization takes place. So this is called isogamy because both the gametes are same in structure. So this kind of fertilization is called isogamy. So due to isogamy, the diploid zygote is formed. Okay, so from this diploid zygote, okay, so due to its continuous binary fission, first it undergoes meiotic cell division, then later uh, its nucleus is divided into mitotically, but here new chambers new chambers are generated. The first chamber is generally known as initial chamber is generally known as prolaculum. Okay, so as the chambers are increased, the number of nuclei is also increased. So gradually, the number of chambers and amount of protoplasm is increased due to feeding. Okay, and it increases in size, and this kind of form of Elphidium with many nuclei is called microspheric form, which is an asexual form. Okay, so this asexual form, after attaining maturity or adulthood, okay, so it starts dividing, mitotically. Okay, each nucleus acquires some amount of, uh, cytoplasm, and so many smaller, amoebulae are generated within the, shell, and they are released outside. They are called amoebulae. Okay, so this kind of amoebulae are produced many in number. Okay, each with a single nucleus. Okay, so now chambers number is increased. This is not nucleus, only single nucleus is present. Okay, and gradually chambers number will be increased, enhanced more in number, and finally it will become a megalospheric form with a single nucleus. So now, due to asexual reproduction, so many megalospheric forms are formed. Here given only one amoebulae is given. But actually, so many amoebulae are produced. From each amoebulae, a single megalospheric form is produced. So microspheric form, which is multinucleate one, undergoes asexual reproduction. Megalospheric form with a single nucleus undergoes sexual reproduction. Okay, now let's see the points. Yeah, the two distinct forms of Elphidium crispum actually display the phenomena of sexual dimorphism. Megalospheric forms are gametes. Gametes and would have listened because it undergoes sexual reproduction. Okay, they reproduce by the formation of gametes called gamogony, which united by isogamy to produce zygotes. Microspheric forms are agametes. They undergo their asexual forms and reproduce by multiple fission called agamogony. Okay, so yeah, they display alternation of generation. This is a clear example for alternation of generation. Yeah, according to Jeffs, the entire cycle of sexual and asexual phase is completed within 2 years. So now let's see. Already we have seen in the diagram. Just I'll read the points. Just wait. Yeah. So gamogamy. First of all, the isogametes are produced. Each gamete means megalospheric form produces numerous identical biflagellated gametes or isogametes. Okay, so the single, so you remember every megalospheric form produces many identical, not one, sorry. So many identical biflagellated gametes which we have seen in the diagram. They are called isogametes because they are similar. The single nucleus divides repeatedly by mitosis, which is accompanied by fragmentation of entire cytoplasm. Each gamete possesses two motile flagella, a nucleus, uh, and a pair of flagella. Okay. Ah, uh, sorry, fat globules and a fat globule, which is for the nutrition. Okay, so such isogametes, they undergo fertilization. The gametes are released into the surrounding seawater through the shell pores. They swim about freely for some time and then elongate to become conical in shape. So before fertilization, they are transformed into conical shape. Later, the gametes originating from different gametes. The రెండు వేరు gametes నుంచి వచ్చినటువంటి the gametes from two different forms, two different megalospheric forms. So they are united and fused in pairs, resulting in a diploid zygote. Zygote అనేది form అవుతుంది. And then agamogamy. So microspheric form or agament, the zygote secretes the shell around a, ఇప్పుడు zygote form అయింది. So it secretes a shell around it, its cytoplasm. A small initial chamber or prolaculum is produced and becomes a young agament or young microspheric form. Okay, so it's diploid nucleus. So I have highlighted this. ఇప్పుడు మనకు మైక్రోస్ఫెరిక్ ఫామ్ is produced, okay, formed, okay, which contains, this is an young microspheric form having single nucleus. Then immediately it divides first by meiosis. ఫస్ట్ ఒక మియాటిక్ డివిజన్ జరగడం వల్ల డిప్లాయిడ్ కండిషన్ ఈజ్ లాస్ట్, హ్యాప్లాయిడ్ కండిషన్ ఈజ్ అచీవ్డ్, అండ్ దెన్ ఈచ్ న్యూక్లియస్ it continues, each one continues mitotic division to give rise several haploid daughter nuclei. Okay, so with further growth and development, new chambers are successively added, resulting in a multinucleate and multilocular adult agamete or microspheric form. It is also, you can also call it as a zygont because it undergoes multiple fission. The nuclei are found in cytoplasm of all chambers and they are encircled by numerous chromatidial granules. Okay, this is not needed for you. The microscopic individuals are scarce in occurrence because of the remote chances of the fusion of gametes. So generally microscopic forms form కావడానికి చాలా తక్కువ అవకాశం ఉంటుంది because so fusion is rare because the ocean is very very big. So schizogony is nothing but the multiple fission, so in which the microscopic form reproduces asexually called schizogony. Entire cytoplasm containing several nuclei creeps out of the shell. Shell నుంచి ఫస్ట్ cytoplasm బయటకు వస్తుంది and takes the form of a lump around it. ప్రతి న్యూక్లియస్ చుట్టూ ఒక సైటోప్లాజం ఒక లంప్ లాగా చేరుతుంది. దిస్ లంప్ నౌ ఆర్గనైజ్ ఇంటూ ఏ లార్జ్ నెంబర్ ఆఫ్ అమీబా లైక్ సెల్స్. ఆ లంప్ నుంచి బయటకు వచ్చిన సైటోప్లాజం నుంచి మెనీ అమీబా లైక్ సెల్స్ అనేటువంటివి ప్రొడ్యూస్ అవుతాయి. దే ఆర్ కాల్డ్ అమీబులే ఆర్ ఏగామెట్స్. సో ఈ ఏగామెట్ అనేది అది నథింగ్ బట్ ది యంగ్ megalospheric form. The agametes now become detached from the parental shell. Each one secretes prolaculum and becomes young gamete or megalosphere. ఓకే, సో ఇక్కడ మైక్రోస్పియర్ నుంచి ఫామ్ అయిన లంప్ నుండి ఓకే మెనీ అమీబులే ఆర్ ప్రొడ్యూస్డ్. ఈచ్ అమీబులే విల్ బికమ్ ఏ మెగలోస్పియరిక్ ఫామ్. దట్స్ ఆల్. సో ఇట్ రిపీటెడ్లీ secretes additional chambers, okay, that will become an adult megalospheric form. Okay, once again I'll show the diagram. Simple, just the diagram ఒకటి గుర్తుంచుకుంటే సరిపోతుంది. So this is megalospheric form with a single nucleus, undergoes sexual reproduction. Okay, so the zygotic nucleus first divides by meiotic division and later mitotic division, produces many nuclei and many chambers are produced to become microspheric form. And each microspheric form, it releases its cytoplasm in the form of a lump of protoplasm having many nuclei. Okay, and each nuclei acquires some amount of cytoplasm to form many amoebulae. Each amoebulae will become adult megalospheric form, which is a sexual form. This is asexual form. Venkatesh, don't touch the screen. Okay, so then this is about the ELPHDIUM crispum. Now, okay, we have locomotion in protozoa. Our next topic is locomotion in protozoa. Okay, so there is no need to study each and every point in this. Some important points you should remember. Some basic points, some fundamental points you should remember. So locomotion is directional movement that enables someone or something to move from one location to another location. We all know the definition of the locomotion. Oh, let's keep it. Yeah. So the word was derived from Latin words loco, place, and motion, to move. And yeah, there are different kinds of locomotory organelles in protozoa or protozoan animals. మనకు తెలుసు pseudopodia are there, flagella are there, cilia, tentacles. Okay, some pellicular contractile structures are also seen. So first of all, let's see about the pseudopodia. What are pseudopodia? Pseudopodia are nothing but an elongated cytoplasm with surrounded by the cell membrane, sometimes an axial supporting filament. Pseudopodia are false feet. They are temporary structures formed by the streaming flow of cytoplasm. Pseudopodia అనేటువంటి they are not, the meaning itself is explaining that they are not true feet. అవి నిజమైన ఫీట్ కాదు, టెంపరీగా ఫామ్ అవుతాయి, మళ్ళీ వెంటనే డిజపియర్ అవుతాయి. కాబట్టి They are called pseudopodia. So there are different kinds of pseudopodia. So here lobopodia, finger-like, philopodia, thread-like, reticulopodia, network-like or mixopodia, mixed one. Axopodia, rod-like or race-like. Yeah. So these are the lobopodia, philopodia, exopodia, and reticulopodia. Reticulopodia, which just now we have seen the reticulopodia in the what we call the elphidium. Yeah, lobopodia, simple. These are lobe or finger-like pseudopodia with broad and rounded ends, as in amoeba. మనందరికి తెలుసు అమీబాలో ఉండేటువంటి pseudopodia is called lobopodia. Club shaped or finger like. They are composed of both ectoplasm as well as endoplasm. Okay. So example, yeah, ఇవి pressure flow వల్ల ఫామ్ అవుతాయి. Example, amoeba and entamoeba. We all know that. And then pseudopodia are nothing but filamentous or thread-like pseudopodia. So such pseudopodia are called philopodia. Filamentous pseudopodia. Example is Euglypha. Okay, they contain a pointed tip. So philopodia are composed of ectoplasm only. Philopodia la matku ectoplasm మాత్రమే ఉంటుంది. అందుకే they are thin. Reticulopodia, just now we have seen. Example, polystomella. Okay, filament are branched which are interconnected. Reticulopodia display two-way flow of cytoplasm. అంటే ఒకసారి ఒకసారి egg ఎలా ఉంటుందంటే I'll draw the diagram. For example, రెటిక్యులోపోడియా యొక్క ఒక చిన్న ఇది అనుకుంటే అందులో సైటోప్లాజం అనేది ఒక డైరెక్షన్లో ఇటు మూవ్ అవుతుంది. ఓకే, ఇంకొక డైరెక్షన్లో సైటోప్లాజం రివర్స్లో మూవ్ అవుతుంది, లైక్ దిస్. సో టూ-వే ఫ్లో. దిస్ ఇస్ నాట్ ఇంపార్టెంట్, waste of time. Anyway, so that is about the reticulopodia. And then axopodia. మనం చూసినాం నిన్న. Can anyone say example for the sun animalcule? Sun animalcule కి ఎవరైనా ఎగ్జాంపుల్ చెప్పగలుగుతారా? నిన్న చెప్పాను నేను. Which animal is called sun animalcule? Anyone? Okay, actinophrys is generally called as sun animalcule. So anyway, they have needle-like straight pseudopodia. Okay, because they contain a central axial rod, which is covered by granular and adhesive cytoplasm. So
అనుకుంటాను నాకు ఈ ఎంకామ్ది ఇంకా ఇంకా పర్లేదు నోటిఫికేషన్ అయితే.
please mute your mic before talking. Okay. So anyway, axopodia means they have needle-like straight pseudopodia that contain a central axial rod. Right. So axopodia display two-way flow of cytoplasm. So their example or they are present in heliozoans. Example, actinophrys, which is commonly known as sun animalcule. And then flagella. So pseudopodia is over. Now flagella. Flagella are locomotory organelle of flagellate protozoa. Flagellata, that means phytoflagellata and zooflagellata. Protozoans, they contain the locomotory organs, the flagella. Example, Euglena and Trypanosoma we have seen. This is phytoflagellate, this is zooflagellate. Anyway, a typical flagellum consists of an axoneme. Axoneme arises from, ఇది గుర్తుంచుకోవాలి మీరు. Axoneme arises from basal granule from the cytoplasm, which acts as an anchor. And the blepharoplast or kinetozoa. Three names are similar, synonyms. Basal granule or blepharoplast or kinetozoa అనే ఒక స్ట్రక్చర్ నుంచి ఈ axoneme అనేది ప్రొడ్యూస్ అవుతుంది. సో ఐ గివ్ యూ, ఫర్ ఎగ్జాంపుల్, దిస్ ఈజ్ a flagellum. If it is a cell membrane, here there is a knob-like structure. So here this is basal granule from which the rod-like axoneme is produced. Actually, it contains many filaments. So this is outer covering, if you assume. Okay, so and if you, so this total structure, which looks like a rod, actually made up of many tubules, that is called axoneme. Okay, so axoneme అనేది సపోర్ట్ చేస్తుంది అనమాట. సో దిస్ ఇస్ అవుటర్ కవరింగ్ ఆఫ్ పెలికిల్, అంటే ఈ యొక్క వాట్ వి కాల్ ది, ఈ పెలికిల్ యొక్క కంటిన్యూషన్ గా చెప్పవచ్చు దీన్ని అవుటర్ కవరింగ్ అనేది. యాక్చువల్లీ ఇలా ఉంటుంది. ఎనీవే, సో ద సెంట్రల్ రాడ్ లైక్ స్ట్రక్చర్ ఈజ్ కాల్డ్ axoneme, రిమైన్ ఎంబెడెడ్ ఇన్ ద ఫ్లూయిడ్ మ్యాట్రిక్స్. ఇదంతా ఫ్లూయిడ్ కలిగి ఉంటుంది. ఓకే, సో దెన్ రైట్, yeah, in axonium 9+2 arrangement is present. So అదేంటో చూద్దాం సరే. So you count it. So this is, just now I have shown you. So this is pellicle continuation. Okay, so this is another membrane called protoplasmic membrane is also present, and this green color one is axoneme. Okay, so it has, so this total structure is axoneme, including this central one. Okay, amma cheppama, is there any doubt? Okay, so anyway, so this one is, this is the TS of a flagellum. Okay, this is the TS of basal granule actually. Okay, so here these are two tubes. Okay, this is one, this is one. Central two tubules are present. Okay, that is two, and you count it. Each one is, each this structure red and yellow is combined known as a doublet. And rendu tubules అనేటివి, as there are two tubules in the peripheral region, 1 2 3 4 5 6 7 8 9. So 9 peripheral doublets are present. Central one pair of singlets are present. So this arrangement is called 9+2 arrangement of flagellar structure. And here, whereas in the, you know, the basal granule, okay, so triplets are present. Each one is a tube-like structure actually. This here, red one is a tube, this yellow one is tube. Such tubes are 3 present in the basal granule, and they are also 9 in number. Triplets, they are called triplets. These are called doublets, and each doublet contains on anterior, you can say, and are A and B. Okay, one of the doublet contains a pair of arms. They are called Dianin arms. So there will be another diagram. See here. See. So these are the peripheral doublets. ఇవన్నీ peripheral doublets. So this one is called A, this one is called B. They are all tubules. Actually, వీటిని imagine this one is a straw. ఇదొక straw అనుకోండి, ఇది ఇంకొక straw, ఇదొక straw అట్లా. So all are tubules. They are not rods. These two are also two tubules which are separated, not attached with each other. Okay, so this structure constitutes the central fiber. Okay, so here this is A, this is B. So out of doublet, such doublets are 9 are present and singlets are 2 are present. This is called 9 plus 2 are flagella. And on A of each doublet contains a pair of an inner arm and outer arm are present. They are made up of a protein called dynein. మీకు పాయింట్స్ లో ఉంటుంది, డయానిన్ ఆర్మ్స్ అంటాం. డయానిన్ is a protein. Okay, and two successive doublets are connected with nexin protein bridge. This is called nexin protein bridge. Okay, so ఇది ఇట్లా కనెక్ట్ చేసేటువంటి పక్కపక్కన డబ్లెట్స్ కలిపేటువంటి the protein is called nexin. And central singlets are surrounded by central sheath. Okay, and each doublet is connected to central fiber with a what we call the radial link or spoke, you can say. In a radial link anta. So madhyalo there is a nodule-like structure called radial link head. So this is the TS of transverse section of a flagellum. Flagellum yokka transverse section, and this is outer membrane. There will be a pellicular membrane is so this constitutes the pellicular membrane. Now let's see the points. Sorry, okay, points. Yeah, 9 plus 2 arrangement. Okay, so, yeah, each peripheral bears double row of short arms made up of protein dynein. So ఇవి అన్నమాట డైనిన్ ప్రోటీన్స్ తో. సో 2 ఆర్మ్స్ అనేవి ఉంటాయి. సో దట్ ఈస్ ఇంపార్టెంట్, వి రిమెంబర్. సో outer arm and inner arm. సో ప్రతిదానికి outer inner they are dynein proteins. They are nothing but the dynein proteins. So types of flagella రకాలు చూద్దాం. I will stop after this. So here also the diagram is slightly mistaken here. Anyway, based on the structures called hair-like structure called mastigonemes, which are present on the either side of the flagella in protozoan, they are called mastigonemes. అంటే ఈ flagellum కి వన్ సైడ్ కానీ టూ సైడ్ కానీ ఓకే చిన్న హెయిర్ లాంటి స్ట్రక్చర్స్ కలిగి ఉంటాయి. ఓకే, సో దే ఆర్ కాల్డ్ mastigonemes. Based on these mastigonemes, the flagella are 5 types. One is stichonematic. That means if the mastigonemes are one-sided, okay, such flagellum is called stichonematic. Example, Euglena and Astacia. You should remember the examples. Panto means both sides. Okay, so రెండు వైపులా mastigonemes ఉంటే, and terminal part is also thicker, just like the remaining part. So such kind of flagellum is called pantonematic. Example, Paranema and Monas. And then acronematic. This is little bit confusing. Simple. ఇక్కడ రెండు ఇచ్చాడు, ఒక్కదాన్ని తీసుకుంటే సరిపోతుంది. So if this is a flagellum, the acronematic flagellum, it doesn't contain any, what we call the hairy like mastigonium, but it contains a terminal filament or terminal hair-like structure or tapering structure or terminal filament which is naked, just like a mastigonium. The terminal part of the acronematic is pointed filamentous structure that is nothing but a single mastigone. Okay, so that is acronymatic and pentacronymatic. Here, just like pantonymatic, both sides mastigonemes are present, and terminal part contains this tapering or naked filament is seen without any pellicular covering. Okay, so here anematic, there are no mastigonemes. Okay, and this diagram is wrong. Actually, it should be like this. So it is very smooth throughout the structure. So that is anematic kind of, flagellum. Okay, example Cryptomonas, pentacronymatic example Arceolus, acronymatic example Chlamydomonas, pantonymatic example Paranema monas. Okay, so once again, let's see the points. Stichonymatic, only one row of lateral appendages are mastigonemes. Present example Euglena and Astacia. And pantonymatic means both sides the mastigonemes are present. Example Paranema and Monas. Acronymatic, lateral appendages are absent, and axoneme, which is a supporting rod-like structure, ends as a terminal naked sharp axial filament. Some believe that the terminal single mastigoneme అని కూడా అనుకుంటారు. Okay, terminal part of the acronymatic flagellum, that is sharp one. Okay, axial filament that is big, that can be considered with a mastigoneme, and single mastigoneme ఉంటుందని అర్థం. So that is acronymatic example Palitoma and Chlamydomonas. And pentacronymatic, both sides, two or more rows of lateral mastigonemes are present, and the axoneme ends in a terminal naked axial filament. ఇది ప్యాంటోనిమాటిక్ దీనికి తేడా ఏంటి అంటే ఇక్కడ రెండు వైపులా మాస్టిగోనీమ్స్ ఉన్నప్పటికీ కూడా సో టెర్మినల్ పార్ట్ ఈస్ నేక్డ్ వితౌట్ దిస్ outer covering. Okay, so this is the anematic without any outer covering. So it is very, very smoother structure. That is anematic, simple without any lateral appendages and a terminal naked filament. Not like this. And move to throughout the throughout the so throughout the flagellum. Okay, so it doesn't contain any covering, and terminal part is more sharp. So example is Cryptomonas. Okay, so here some examples with flagella. Euglena has two flagella. One is short, another one is long. Trypanosoma has one flagellum. Trichomonas has four flagella. Giardia lamblia or Giardia intestinalis. Yesterday I told you 8, uh, you know, uh, 4 pairs of flagella. And Trichonympha contains many, many flagella. Trichonympha. So that is about today's class.